For most of human history, a sweet taste was incredibly rare. Finding a piece of fruit or wild honey meant you had secured safe, high-energy carbohydrates. To make sure we ate as much of it as possible, our brains evolved to reward us with a massive hit of dopamine. But today, sugar is everywhere. Our biology hasn't caught up to a food environment defined by constant, chronic overconsumption. This diagram maps out exactly where that excess leads. Overlinear system creates a direct biological pathway to insulin resistance, type 2 diabetes, non-alcoholic fatty liver disease, and severe cardiovascular issues. This leaves us with a severe nutritional paradox. How do we keep the deep neurological pleasure of sweetness, but eliminate the metabolic damage caused by the sugar itself? Food scientists found a solution by isolating the target. This illustration shows the T1R2, T1R3 receptor complex, which acts as the body's primary gateway for perceiving sweetness. They discovered a loophole in human biology. You don't need sugar to activate this pathway. Entirely different engineered molecular structures, like saccharin or brazine, can be shaped to bind perfectly with this same receptor site. Once they bind, they send a signal straight up the neural pathway to the brain's gustatory cortex. You perceive a rush of intense sweetness, even though there are no carbohydrates or calories present in your stomach. By triggering this specific biological response, we believed we had outsmarted our own evolution. We engineered a way to get the reward of sugar with zero metabolic cost. To understand how we deployed that hack, we have to look at the tools. This chart breaks down the major categories of engineered sweeteners, starting with pure synthetic compounds. Take Advantame, highlighted here. It is synthesized to be 20,000 times sweeter than regular sugar, but extreme potency creates a physical problem. Because you need such microscopic amounts of the chemical to taste it, these synthetics provide no physical bulk, weight, or texture to the foods they're put in. To fix that missing structure, food scientists use polyols. Maltitol, seen in the center panel, mimics the physical weight of real sugar, making it essential for baking. But consuming the large volume needed to mimic sugar crosses a digestive threshold, triggering severe osmotic distress. The third strategy tries to bypass synthetics entirely. Driven by consumer demand for clean labels, companies extract botanical compounds like stevia. Yet, as this breakdown shows, these naturally derived molecules carry bitter aftertastes that make them difficult to use on their own. Every alternative molecule we have found carries a major physiological compromise. A perfect, solitary one-to-one replacement for sugar does not exist in nature or the lab. So, the industry adapted. Modern zero-sugar products use an interlocking network of chemical compounds to balance out each other's flaws. By combining cyclamate and saccharin at exact ratios, their structures effectively cancel out each other's harsh metallic and bitter notes. Recreating the simple, clean profile of regular sugar requires an elaborate, fragile chemical balancing act. For decades, artificial sweeteners were considered inert. The traditional view believed they passed cleanly through our digestive tract, but emerging science reveals these zero-calorie compounds actively alter bacterial adhesion in the gut, promoting pathogenic biofilms. The irony is severe. By disrupting the microbiome, these completely carbohydrate-free sweeteners can actually induce the same glucose intolerance they were intended to prevent. The damage reaches past our bodies. This environmental cascade shows that the extreme chemical stability of these synthetics allows them to survive wastewater treatment entirely intact. Accumulating in plant nectar, they interfere with the chemosensory perception and memory of vital insect pollinators. Our attempts to outsmart our human metabolism triggered an unintended cascade of damage, affecting both our internal microbiomes and our external ecosystems. Despite these consequences, we can't simply abandon these compounds. Looking at these market statistics, a massive global diabetic population relies on this multi-billion-dollar industry for baseline health management. But the method of formulation must change. The search for a single, universal, healthy sugar is based on a flawed premise. Future research is moving toward the individualized model shown here, often referred to as precision nutrition. We are moving away from universal formulas, focusing instead on tailored chemical profiles based on an individual's unique genetics and specific gut bacteria. We have not yet solved the paradox of sweetness. But by moving past the hope for a simple biological freebie, we have finally begun to learn the complex rules required to navigate it safely.